All right, so let's dive into this drug development stuff. You sent some fascinating sources about what makes a molecule drug-like,
Hmm.
and a lot of it seems to have something to do with ADME.
Yeah, ADME, it's a pretty fundamental concept. Stands for absorption, distribution, metabolism, and excretion.
So basically, what our bodies do to a drug once it's inside.
Exactly.
Yeah.
It's figuring out the journey a drug takes once it enters the system. Makes sense to study it early on,
Oh, yeah,
too.
for sure. You wouldn't want to spend years developing a drug that just disappears from the body immediately.
Right. Or even worse, turns into something harmful.
One of the sources mentioned metabolic stability as being super important.
Yeah, so during lead optimization, they're screening those drug candidates, checking how they break down and how long they stay in the system.
Like a molecular obstacle course.
I like that. A lot of it is done in vitro too, outside a living organism.
So not in like a person or an
All
animal.
right, they use things like liver microsomes.
What are those?
Think of them as mini livers in a test tube.
Wow. So they're recreating what happens in our bodies, but in a more controlled environment.
You got it. It gives them a sense of how stable a molecule is if it breaks down too fast. Well,
It's back to square one.
pretty much.
Yeah.
But one of the articles mentioned that sometimes a drug breaking down quickly can actually be a good thing.
Really? That seems counterintuitive.
It comes down to the metabolites,
Which are?
the products that are formed when a drug breaks down.
Okay.
And sometimes those metabolites are pharmacologically active themselves.
You mean they can have effects just like the original drug?
Sometimes even better therapeutic properties than the original molecule.
Like a bonus price.
Exactly. But there's also that a darker side to metabolism too.
Right. Something about reactive metabolite.
Unfortunately, yeah. While some are helpful, others can be problematic.
Problematic how?
They're unstable, and they can interact with our cells and cause damage.
So that's where you get those side effects and toxicity. Liver toxicity, DNA damage, triggering immune responses. That's why understanding what those byproducts do is so crucial. Catch those red flags early.
I see. It's amazing how a drug just breaking down can have such a huge impact.
It's a delicate balance.
So how do scientists even predict whether a molecule will be absorbed well in the first place with all these different aspects of ADME?
Well, they use certain guiding principles. One of the most famous is Lipinski's Rule of 5, kind of a cornerstone of medicinal chemistry for decades now.
Lipinski's Rule of 5.
Yeah.
Okay, what's the significance of the number 5?
Not magic, but it's based on observations about the characteristics of successful drugs. So for a molecule to be absorbed easily, it shouldn't break too many of these rules. Molecular weight less than 500, no more than 5 hydrogen bond donors, no more than 10 hydrogen bond acceptors,
Right.
a certain level of greasiness, which is measured as log P, and that should also be less than 5.
Okay, so these are like guidelines to predict how well a drug will get into the bloodstream.
Exactly. It's a filter for weeding out molecules that are unlikely to be absorbed well. It might have great activity in the lab, but if it can't even reach the bloodstream...
It's not going to be very useful.
Right. So are there exceptions to this rule? Sure. Especially with all these new therapies coming out,
You're talking about things like biologics.
like antibodies and PROTACs, which work completely differently. Lipinski's rule doesn't always apply.
So these newer drugs are playing by a different set of rules, though. What makes them so unique?
Biologics, for instance, they're much larger molecules than the typical small molecule drugs that Lipinski was thinking about. They work by binding to very specific targets, usually on the surface of cells.
Interesting. So it's less about being absorbed into the cell itself, but more about interacting with it from the outside.
Exactly. Highly specialized keys fitting into specific locks on the cell's surface.
Mhm.
And their size, which would normally be a disadvantage according to Lipinski, actually works to their advantage.
Fascinating. What about PROTACs? How do they bypass these rules?
PROTACs are really interesting. They act like molecular matchmakers. They bring together a target protein and the cell's own protein degradation machinery,
It's like hijacking the cell's natural cleaning system.
a Trojan horse.
Exactly, to get rid of those harmful proteins.
And because they're designed to be recycled within the cell, they don't need to follow the same absorption and distribution rules as conventional drugs.
I see. So they work from the inside out.
You got it.
This is amazing. Drug development is really pushing the boundaries.
Yeah.
But even with these exceptions to the rule, scientists still need to understand how these new types of drugs are distributed, metabolized, and eliminated from the body.
Of course.
Hmm.
It's not a free pass just because they're unconventional. You need the whole picture. How it gets in, where it goes, how it breaks down, how it leaves. And that's where those early ADME tox models and high-throughput testing come in.
Okay, so tell me more about that.
Well, that's a whole other story. Yeah, it's not a free pass. You're right.
Yeah.
You still have to understand those key aspects, even with these groundbreaking drugs.
So how are scientists tackling this? Especially early on in development. You mentioned ADME tox models. What are those?
They're like a sneak peek into the future of a drug candidate. You get a glimpse of how it might behave in the body, way before human trials.
So like a crystal ball for drugs. But how can they predict what will happen in something as complex as the human body?
No model is perfect, but they give you really valuable insights. Some of them use computer simulations. They're getting so sophisticated now.
So it's like those personalized recommendations you get online, but for molecules.
Haha, kind of.
Yeah.
They consider so many factors, potential drug interactions, genetic variations. It's pretty amazing.
That's incredible. What about those mini organs I read about, the organoids? How do they fit into all of this?
Organoids are really changing the game. They give us a more realistic system to study drug effects compared to traditional cell cultures.
So it's like a miniature version of an organ in a dish.
Exactly. You can actually see how a drug is absorbed, distributed, metabolized, even assess its toxicity. It's a much more accurate picture.
Wow, that's pretty sci-fi. Sounds like these models are really bridging the gap between the lab and the real world. But even with all these advances testing, each potential drug must still take a lot of time. Right. You mentioned high-throughput testing earlier. How does that speed things up?
Imagine a bunch of tiny robots working nonstop, testing thousands of compounds at the same time for very specific properties. That's high-throughput testing. It really accelerates the screening process, especially for ADME.
It's like a cooking show with tons of ovens going at once.
Yeah.
And instead of food, they're making data.
I like that visual. And one of the key things they test is permeability. How easily a drug can get across cell membranes. It's crucial for it to reach its target.
It's the ultimate test for these molecules.
And with high-throughput testing, scientists can quickly identify the winners, the molecules that can be absorbed well. They weed out the ones that don't make the cut.
So it's about being efficient and strategic with your testing.
Exactly. And
Yeah.
by combining these advanced models with fast screening methods, it really streamlines the whole drug development process.
It's amazing how far things have come. What are your thoughts on where this is all heading?
It's a very exciting time to be in this field. It's not just about speed, it's about precision too. We're moving away from that one-size-fits-all approach to medicine and tailoring treatments for individual patients based on their genes and other factors.
So personalized medicine, but on a whole other level.
Exactly. And understanding ADME is at the heart of this whole shift. If we can predict how a drug will behave in different people, we can develop safer and more effective therapies.
That's incredible. We've covered a lot today. Anything else that you think is important for our listeners to take away?
I'd emphasize the importance of research and innovation in ADME. As we make new types of drugs and learn more about the human body, our understanding of ADME has to keep up.
So it's not a static field. It
Yeah.
needs to keep evolving.
Yeah.
Just like drug development itself.
Exactly. And this ongoing research will lead to better models and testing methods, refining our ability to predict how a drug will behave in the body. It's about minimizing risks and maximizing the chances of success in drug development.
It's fascinating how something seemingly simple, like what our bodies do to a drug, can be so complex.
Yeah, it really shows how much teamwork goes into drug development. It's not just one person in a lab. It's chemists, biologists, pharmacologists, even computer scientists,
Yeah.
all working together.
It really does take a village, and ADME is such a critical part of that whole process.
Because at the end of the day, it comes down to that one question: will this molecule actually be a good medicine?
Exactly. And it's not just about showing it works in the lab. You need to understand how it's absorbed, how it moves through the body, how it's broken down, how long it lasts, and are those breakdown products safe?
It's like tracking the molecule's journey through the body, with all its twists and turns.
And the more research we do, the clearer that picture becomes. That's leading to some really amazing advancements in medicine.
Safer, more effective, and more personalized treatments.
Exactly. It has the potential to really revolutionize healthcare. But there are still a lot of questions that need to be answered.
What would you say are some of the biggest challenges in ADME research right now? One area that's really exciting is the role of the microbiome in drug metabolism.
The microbiome. You mean the bacteria that live in our gut?
Exactly. We're just starting to understand how those trillions of bacteria can affect how our bodies process drugs.
So it's not just about human enzymes anymore.
Right. It's a whole ecosystem in there. And it can vary a lot from person to person. So scientists are trying to figure out how these microbes contribute to drug metabolism and whether we can use that knowledge to make drugs even better and safer.
That's fascinating. It's like our bodies are these incredibly complex worlds with so many factors influencing our health.
It really is. And the more we learn, the more we realize how much we don't know.
It sounds like there's still so much to discover. Well, I think we've covered a lot of ground today, and it's time to wrap up this deep dive.
Yeah, we've only scratched the surface of this really complex and constantly evolving field.
But hopefully our listeners have a better appreciation for ADME and how important it is in drug development.
Absolutely. Those questions about absorption, distribution, metabolism, excretion, they might seem simple, but they have huge implications for the medicines of the future.
It's been a really fascinating conversation. Thank you for joining us today.
It was my pleasure. Thanks for having me.
And to all our listeners out there, thanks for tuning in. We'll be back soon with another deep dive into the world of science and technology.